Hello and welcome. My name is Mitch Sanders. He is Jordan Yarn. We're honored to have another Wound Healing Society Awardee Highlight today with Kanhaiya Singh. And welcome. How are you, sir?
Thank you. I'm good. How are you?
Wonderful. Kanhaiya is a associate professor at the University of Pittsburgh. He actually did his doctoral degree at Banaras Hindu University in India, did his postdoctoral research in epigenetics at Ohio State, and was assistant professor at Indiana University Department of Surgery as well. And then you moved to Pitt, and you're doing amazing science that we're going to talk about today. Anyway, we're going to talk about MIR200B and how looking at ischemic mice and how that regulates so many different proteins. And if you can downregulate MIR200B, then you have a chance of getting VEGF to stimulate angiogenesis. So could you first talk about your career and how you got into wound care, and then talk more about the science that was highlighted in the award that we gave you that last year?
Thank you, Dr. Sanders. So, as you mentioned, I started my PhD or graduate study back in India. And that time onwards, I started doing research on wound healing, especially on diabetic wounds, because as you know, right now, around 800 million patients are suffering from type 2 diabetes. And roughly around 25% of those develop some chronic wounds which will not heal. And it has been noticed that around 1/4 of those people who develop wounds will ultimately leads to amputation. And amputation is very high in mortality rate. Around 50 to 60% of patients having amputation due to DFUs will die in 5 years. So that is the extent we are looking at. It can be compared to some kind of cancers where the mortality rate is not that much high too. So this is the motivation why I started looking into molecular and epigenetic mechanism of diabetic foot ulcers. And what my lab believes is, in addition to genetics, diabetes is an epigenetic disease. That means not only the genes that are responsible for development of the chronicity, the environment also have an important role to play. And by epigenetics, I mean that any changes in gene expression, which is not governed by the DNA. It is governed by some mechanism which is above DNA. For example, DNA methylation, histone modifications, and microRNA regulation. So this paper that got a translational award from WHS was looking at a microRNA called microRNA 200B and its regulation in wound angiogenesis. So this was the premise of the work, and that's why we started looking into MIR200B. So microRNA, if you define microRNA, it's a small RNA molecule around 20 to 22 nucleotides long, and they can regulate mRNA expression by either binding to the mRNAs and cleaving them, or making them inaccessible for ribosomes to make proteins. So, and microRNA 200B, when we reported, our lab reported back in 2011, that it is an angiostatic microRNA. That means if it is there, the blood vessels that needs to form in order to supply blood to any ischemic tissue will go down. So through a series of experiment in mice, in porcine, we saw that if there is an injury, the level of microRNA should go down with progression till proliferative phase. At the same time, when microRNA 200B is down, the rate of angiogenesis increases. And once the wound is healed, the microRNA 200B levels come back, and then angiogenesis it also need to regress, because if you have formed good vessels, you don't need sustained angiogenesis phenomena over there. You need homeostasis. So microRNA level also play with the homeostasis of newly formed blood vessels. So, but when you see diabetic wounds, this kind of downregulation is not observed. MicroRNA 200B remains high in wounds of diabetic individuals. So we are looking into why these levels are remaining high, and if by any therapeutic measures we can reduce the levels, what happens to diabetic wounds. So a paper that we published in 2017 explained that not only microRNA levels remain high, it is high because its DNA methylation goes down. So it is the promoter of microRNA 200B remains hypomethylated in diabetic wounds. And if you can by any mean like rescue the methylation, you are able to like bring down mid-200B levels and bring angiogenesis up in those diabetic individuals. So based on those previous findings, now our lab is looking into that what is the contribution of microRNA 200B in different compartments of wounds. For example, uh, fibroblast, what it is doing in fibroblast compared to what it is doing in endothelial cells. So when we looked into fibroblast, we found that if you somewhat decrease microRNA 200B from fibroblast, they will start acquiring vasculogenic properties. But at the same time, they will not lose their fibroblastic properties. You know, so that means you are having a fibroblast which is behaving like a endothelial cells. And with single cell RNA sequencing, we noticed that not only VEGF pathways are high in those fibroblasts, but also COL1 and every other collagen producing genes are also there. So we termed them as vasculogenic fibroblasts, VFs, and that was reported in 2023. So, but you know, these are very transient events. Fibroblasts need endothelial cells and endothelium to integrate and form a functional blood vessels. So that means your endotheliums will also respond to such microRNA 200B regulation. So now we are finding through single cell RNA sequencing that when you diminish microRNA 200B or elevate microRNA 200B in an in vitro model, you are able to get few clusters of endothelial cells that are MIR200B responsive. And we are now looking into that what clusters they are, what are the genetic profile of those cells, and why can't we mimic those profiles inside in vivo condition, and how can we rescue the perfusion that is lost in diabetic limbs. So for that, we have now developed a mice which has a flux guiding microRNA 200B and its sister microRNAs. And then we cross it with a Cree, which is a endothelial Cree called Tie2 Cree. So that means when this animal is made in endothelium, microRNA 200B is down genetically. That means we are not biasing our approach over there. So if microRNA 200B remains down in those individuals or in those mice, and if you provide hyperglycemic conditions to them by streptozotocin, you are seeing a good response in those microRNA 200B deficient mice compared to wild type mice, that when they are hyperglycemic, their ischemic limbs are better perfused when microRNA 200B is not there in endothelial compartment. So this is in a nutshell of what we have done so far. And we are still working on that project, and we are trying to see that how fibroblast and endothelial cell crosstalk happens and what is going on in those individuals, in those animals.
It's so cool.
I'm gonna jump in with a question because I'm really excited by this work, and because I only have 5 minutes before I have to hop off. But I mean, I've followed your work long before I met you, and one of my favorite papers of yours was the 2019 paper looking at glycemic state in ZEB1 and its regulatory effect on angiogenesis. Of course, some of this work was published a couple years ago in Nature Communications, and I'm curious, to what degree do you see sort of echoes of the earlier ZEB1 study? And here, is there a glycemic state driver to this elevated level of miR200B that might eventually, lead to breakdown of angiogenesis, breakdown of epithelial barriers and ulceration and things like that, even without wounds? So are you looking into this effect of glycemic control of miR200B and whether it has that effect of causing a sustained high level?
Yeah, so nice point. So ZEB1 project we started because ZEB1 is a known target of microRNA 200B. And when we looked at ZEB1 in non-diabetic animals, we found that the hypothesis is still valid in those like in normoglycemia. When ZEB1 is down, angiogenesis is down because miR200B is supposed to be up because it has a feedback regulation also. ZEB can regulate microRNA 200B. But when we looked into hyperglycemic individuals or animals, we found that ZEB1 levels was high in those individuals too, because ZEB1 functionality is lost during hyperglycemia. That means it is not able to bind to the molecules it should bind, and that's why molecular machinery will keep on producing more ZEB1. So
you have a great chip.
So even ZEB1 levels was high, but it was non-functional.
Yeah.
And same thing is now we are seeing what is happening in fibroblasts. At that time, we didn't have single cell level resolution. Now we have single cell level, and now we are trying to see that ZEB1 levels are down only in endothelial cells or up, or the other cells are compensating it. And we found that, yes, there are other cells like fibroblast and epithelial cells, which are compensating the ZEB1 levels, which is down in endothelial cells. So this is somewhat related, microRNA 200B ZEB axis, and not only angiogenesis, it also controls epithelial to mesenchymal transition. That means the animals having less ZEB1 will have lesser barrier function.
When we see each other at the end of the month, I think we'll see each other. I'll tell you about how that one paper inspired probably the last 5 years of work in our group. So it's I want to pick your brain about it in Texas later this month.
Sure, we will meet.
So I have another question, just briefly. I know we don't have a ton of time, but what we found with bioactive glass, and it's a paper we're submitting this week to AWC, it actually indirectly stimulates macrophages through a bunch of kinase cascades, and also directly interacts with endothelial cells. And based on gene expression studies using nanostring, we saw 5 genes downregulated, 24 and 19 upregulated, and they're all involved in the angiogenesis. And what I was so amazed at, as you talk about vasculogenic fibroblasts, I'm wondering if there's a role that macrophages play, at least for priming and getting the endothelial cells ready by producing more VEGF and all these other chemokines and cytokines that can stimulate endothelial cells as well. And we're also looking at one of the genes we're looking at is causes DNA methylation as well, and we also saw enhancers of like VEGF-A and VEGF-C playing an important role in this whole process. So what's really cool is you could start to tease out the kind of direct interactions of these different cell types in causing, angiogenesis and how. But I was just amazed that MIR-B can act on 319 proteins. So it's I'm only seeing a very when I do nanostring, I only look at like 300 genes at a time. You're looking at a whole transcriptome, looking at doing sequencing of the whole cell. So you're coming up with just the data is just amazingly rich for years to come. What do you think you want to do in the future after? What are the next steps for this work?
Yeah.
I'm gonna hop off.
Yeah.
Please continue, and
Yeah.
I'll watch the video to hear the rest of this answer. Thank you.
Thank you.
Great to see
Yeah,
you.
to echo your point that myeloid cells or macrophages, we are doing single cell sequencing on those macrophages from wounds and others, and we are finding that macrophages are made up of 4 or 5 different cellular clusters, subclusters.
Yep.
And 1 or 2 of those clusters are actually pro-angiogenic, and 1 or 2 are anti-angiogenic, and this is the ratio that is creating the things. And what we are doing right now, we are taking out those pro-angiogenic clusters, looking at their molecular profiles, and then trying to mimic that in vivo systems, so that we can how we can increase the pro-angiogenic macrophages to support the vascular compartment over there. So one of my NIH funded study, we are looking into the endothelial clusters and macrophages cluster, and we are looking into the crosstalk. So what we found that endothelial cells secrete a factor called phospholipase gamma, which binds to TREM2 on macrophages, and this creates access to form new blood vessels at the site of injury. And those TREM2 expressing macrophages or myeloid cells are basically anti-inflammatory in nature, maybe similar to M2 phenotype.
Yeah, exactly.
So, yeah, so of course, this is one of the aspects that we are looking into, that what happened to these MYR200B deficient endothelial cells, how they connect themselves to macrophages, especially the pro-angiogenic macrophages, and fibroblast, especially the vasculogenic fibroblast. So this is in nutshell what we are going to do in future.
So I hope you don't mind. I would love you to be a reviewer for my paper that I'm submitting.
Of course, I will be happy to.
No, that's wonderful. So tell me where you're going to go next with the research. What do you foresee in the next couple of years for your lab?
Yeah, so in next couple of years, what we are trying to do, the lessons that we are learning through single cell RNA-seq and epigenetics, we are trying to develop a dressing that can, in timely fashion, can deliver VEGF and those epigenetic principles temporally to the wound site. So this is what we are thinking right now. And these days you might have heard about micro needles, micro patches, bioscaffolds. So this is how we want to do some preclinical and clinical work to establish the clinical significance of our in vivo and in vitro experiments. So this is the near future that we are looking at.
That is so cool. We've done a lot of preclinical work, but we're now doing clinical testing, trying to understand the mechanism, whether the mechanism action in the mouse matches, a diabetic foot or a venous leg ulcer, what have you. So I think we're all going hopefully in the same direction, which is so exciting. I can't wait. The corollaries with what you're doing and the paper we just wrote is just it's just so interesting and fun and different. And I have to congratulate you on the wonderful work that you've done for your scientific career. And we're so delighted to see you at SADWC WHS coming up this spring in a month. Oh my gosh, things happen so quickly.
Yeah, I'm WHS. I want to thank WHS for these opportunities every year that we get as young researchers, and then as you develop. So like few years back, I was competing in young investigator uh, uh, competition that WHS has, which is one of the most fierce competition that we have. And now throughout the career, now to this WHS, I have a session called Meet the Mentors. So, so I dedicate this success to WHS, the Wound Healing Society, how to shape a career of a young investigator into an early career investigator also. So, and with this also, I will like to thank the sponsors that are sponsoring my lab, especially NIH, DOD, and Commonwealth of Pennsylvania, so that my lab is doing what we can do uh, based on the fundings that we get.
Amazing work. You also had two posters in diabetes that relates
Yes.
to this work as well. Are you when is this work gonna? It sounds like there's 3 or 4 papers that's gonna easily come out of this work. What are the next papers coming out?
So we are one paper which is basically dedicated to single cell differences in diabetic wounds and severe diabetic wounds. Right now in pipeline. The other paper, this paper that we just talked about, microRNA 200B and endothelial compartment and how it crosstalk with other fibroblast and myeloid compartment. So these are the two like near complete papers that we are targeting. There is another paper which is supported by DOD on a different note, but it is also how we can enhance VEGF therapy outcomes by demethylating a gene of interest called metallothionein. So we are trying to demethylate it and we are trying to rescue VEGF therapy. So yeah, so few papers in line, pipeline right now,
Wonderful.
and hopefully we will be doing that. Yeah, on ADA, I am now serving on their membership advisory group.
Oh, great.
So I am like one of the person who vouch for foot care basic science research in their committee. So that I'm proud of right now.
That's
So
wonderful.
hopefully the research portfolio of ADA, especially dedicated to diabetic foot ulcers, I can contribute to in future using that platform.
Anyway, it's great to see you. I can't wait to see you down in, what is it, Grapevine, Texas, in a month. And
Yeah.
thank you for being such an amazing scientist. We love following your career. Anyway,
Thank you very much.
thank you kindly.
Bye.
Bye.